Everybody. We are talking about electrophoresis in flatbed systems. Why? Because a lot of people, when they think about protein electrophoresis, for instance, mainly think about vertical systems, small mini gels in cassettes and things like this. But parallelly, during the development of all these polyacrylamide gel electrophoresis methods, there has always been also a development on horizontal flatbed systems. And I want to show you that the flatbed systems have a lot of advantages which you might be interested in. You see here already the latest developed design by Serva. This is a horizontal chamber. I will explain it a little bit more in detail later on, where you can run gels with different methods, electrophoretic methods. The timetable for this short presentation will be: first we compare horizontal systems and horizontal gels to verticals. We briefly talk about environmental aspects, then about the instrumentation, also what's new, maybe very useful for you. Then about rehydratable gels, the clean gels. When would you need this type of gels? Especially about isoelectric focusing, about immobilized pH gradients, and finally about 2D electrophoresis also in flatbed systems. Yeah, first the comparison. You see a small drawing here, how typically a gel can look like in a flatbed system, and here how a ready-made vertical gel cassette, gel in a cassette looks like. So I read out first about for the flatbed systems. So you can use flatbed systems particularly for isoelectric focusing, and for IPGs, for immobilized pH gradients, you almost have no other choice. The gel thickness, however, is limited because you can cool only from one side. But on the other hand, you cool very efficiently because you place the gels, usually cast on a film support, on a thin film support, plastic film support, directly with a cooling plate. So you have no indirect cooling. That is very efficient. Also, it is very versatile because with one system, with one instrument, you can run different gel sizes and different methods very easily. And the gels, as I said, are mostly cast on plastic films. So if you have only a few samples to run, you just can cut off part of the gel and store the other part in the refrigerator. In the vertical system, have the benefit that you can run more gels at one time. So there are systems for 2 gels, 4 gels. There are also systems for 6, for 9, or for 24 gels at a time. Mostly it's dedicated for all types of zone electrophoresis, like SDS electrophoresis, DNA fragments, and native electrophoresis. Also, another benefit of vertical systems is that there are thicker proteins available. So you have a higher protein loading capacity if that is needed. And you can cool the gels from both sides. On the other hand, I have to say the cooling from both sides is usually indirect. So you cool first the buffer, and then the buffer will cool the glass plate, and the glass or the plastic plate, and the plastic plate cools the gel. So it's not as efficient like for the flatbed system. Further on the flatbed systems, you are usually not using buffer tanks anymore. So you are running them with buffer wicks, and so you don't need large volumes of liquid buffers. And for the 2D polyacrylamide gel electrophoresis, you can also use thin gels, and thin gels have the benefit of higher resolution, sharper spots and sharper bands. Here is no problem to apply an IPG strip because you have an open surface. But you need, but also I have to say you can but you need to remove the IPG strip after the proteins have left the IPG strip and are completely transferred into the second dimension, because otherwise you could get some troubles or problems with electroendosmosis. If you need more explanations on electroendosmosis, please check on YouTube. We had also some basic webinars where we explain this very thoroughly. In vertical systems, you have the gel size is fixed by cassette size. So one system is only fitting for special cassettes. And you cannot easily use thin gels because the IPG strip has to fit into the cassette. And so therefore the gel cannot be thinner than 1 millimeter. Otherwise it would not fit into the cassette. And you don't remove the IPG strip during the run. This has the benefit that you don't need to interrupt the run to remove the IPG strip, but it has the disadvantage that the IPG strip will stay there, and IPG strips have fixed charges which influence the electric field, so you can get some little distortions by it, which is not the case in a flatbed system where you remove the IPG strip. So how do the gels look like? For isoelectric focusing, there are gels available which are just used like this. You apply the samples with sample pieces or with some strips, loading strips, which have silicone strips with holes in it. In many cases, isoelectric focusing is done with electrode strips, which are soaked in an acid at the acidic side, and with a base in the cathodal end, side. There are also gels available which contain sample wells for isoelectric focusing. For zone electrophoresis, we need always sample wells, otherwise the pattern is not really good because it has some stacking effect there, the sharpening effect. Formerly, the people were using 1 liter buffers in tanks, connect the gels via these electrode bridges, usually paper material, wicks material. But you can also use buffer concentrate in polyacrylamide gel strip, or, which is the most easiest and nicest method, is just to soak fabric wicks like a fleece material in the buffer concentrate and place them on the 2 edges of the gel, and then this is easy to dispose of. So horizontal gels have some advantages which I will show you here. So first of all, here you can make gels with very high throughput. With many sample wells, you will see a gel with 103 sample wells. So it's no problem to do that in one gel. They are usually on film backing. You can cast these gels yourself, but you can buy the gels. And film backing has the benefit that it mechanically supports also gels which are very thin, which have large pore size and thin at the same time, so the gels cannot break anymore, and the gels are easily dried down on the films for archiving as a document, as a real document. So it makes life easier. You don't need any cassettes. You just take the gels out of the package and place them on the cooling plate. You need low buffer volumes because you usually use buffer concentrates on both sides, so it makes life also easier. And very thin gels have higher resolutions than thicker gels. This has been proved by many papers already. I'll show you one in the end. And on a horizontal system, it's easy to apply high voltage because you have the electrodes arranged sideways. In vertical systems, the electrodes are dipped into the buffer with a larger volume, and there you are restricted to maximum voltage. Also, the temperature control on these thin gels on film supports directly on the cooling plate is much better than indirect cooling or temperature control, and it is you can achieve very efficient heat dissipation. This is particularly needed with isoelectric focusing with carrier amphiolites, because across a pH gradient you have different conductivities. And in areas of low conductivities, where the carrier amphiolites are not so concentrated, you will get very high electric fields in these points. And that means that the gel can heat up in these little areas quite high, and when the heat dissipation is efficient, the gel will not burn through. Otherwise you can have problems with gels burning. You have a wide choice of buffer systems. I will go in more detail in a few slides later. And this is also an important point, you have the possibility of rehydrating the gel with urea. So denaturing conditions are, in many cases, needed for isoelectric focusing, but there are also applications where you really need urea in the gel. So then you have to cast the gels short before use, or when you want to use ready-made gels, you don't want to work with acrylamide monomers and all this, you can use readily polymerized and dry gels and just soak them in urea solution. In a short time we have some very good working system. And we should not forget environmental aspects. I have in many, on several of my seminars at least, had some comments from people. They were criticizing the ready-made vertical gels. You know, there's several companies supplying ready-made vertical gels in cassettes, but these are producing a lot of waste, so in a horizontal system you don't have any waste except the packaging, but the packaging is always needed also for vertical gels in order to avoid drying out of the gels. Ready-made gels on film supports don't need cassettes, and you have, as you see, low buffer volumes, easy to dispose of, particularly when you work with some problematic samples, serum and so on. And the cleaning requires only small amount of water in a big boxes with big buffer tanks. This is not so easy. You need a lot of washing with water. Yeah, here you see the setup, at least part of the setup of the HPE BlueHorizon flatbed chamber. So there is the power supply. You can, as you see, plug in up to 4 chambers which you might run parallelly. What you do not see on this picture, the instrument here is connected to a chiller, to a cooling machine, a thermostatic circulator, which is pumping water with a defined temperature to the cooling plate. Here, this drawer is the cooling plate. This is aluminum oxide ceramics, which has a very good temperature transfer rate, much better than glass. And what you see here, this transparent plastic, this is the electrode lid, which is now sitting in its parking position. So in contrast to former systems and horizontal system, electrophoresis, the flatbed apparatuses, all this can be placed on one little footprint area, including also the power supply. And the old systems needed a lot of space because you had a separate lid and then a separate electrode holder plate and things like this, where you need space for it. Here's everything concentrated in a small area. You can use these systems, the gels, for native polyacrylamide gel electrophoresis, SDS page for isoelectric focusing, including 2D electrophoresis, high resolution 2D page. I don't want to forget to tell you that a lot of these type of gels are available from Serva. So Serva offers SDS one-dimensional gels with different gel concentrations on film supports. Then CleanGels, these are dry gels which are rehydrated short before use. I'm going to explain this shortly for you. And then there are the Serva pre-coats. Several pre-coats are very thin polyacrylamide gels, which are available with 150 microns or with 300 microns, all polymerized on plastic film, readily available for the run. Then there are the focus gels. The focus gels are very special. They are pre-polymerized, washed, dried down, rehydrated again, and they are thicker. They are 650 microns, and they are particularly designed also for samples with higher salt protein loads, because they have a higher loading capacity than pre-coats. And then there are the immobilized pH gradient strips, which are almost exclusively used for high resolution 2D electrophoresis. And finally, the high performance, the HPE Electrophoresis 2D gels, which I will show you also in a moment. First, some SDS Polyacrylamide Gel Electrophoresis, where you get a separation according to the molecular size. Here is an example of a ready-made gel with 10%, 25 sample wells. This is meant by the 25S. It's 0.43 millimeter thin, contains sample wells, as you see here, for up to 15 microliters sample. And an important point I forgot to tell you is that in vertical systems, you have to underlay the sample with glycerol, and here you don't need this glycerol in the sample. You can directly load the sample to the sample well, because you don't have buffer on top of it. Here you see size markers. Here is human serum, is the albumin, for instance, the IgGs. Then there are E. coli extracts, horse muscle extracts, just to see to show you some different types of samples. And the staining here was performed with Serva Blue R staining kit, which is also readily available. I think you see that we get very nice protein pattern with these gels. Another one is a 12.5%. NF means non-fluorescence. Meanwhile, all these SDS gels are on a non-fluorescent film support. Then you can use fluorescent protein staining and also fluorescent protein labeling. There is the trend to fluorescence detection for staining and labeling, because it is much more useful for quantification, and it makes life much, much easier, and is another aspect of environment friendliness when you first just label the samples prior to the separation, then you don't need staining solutions anymore. For this, you can also check one of our other webinars on the detection of proteins. And here you see marker proteins, HeLa cell extract, and E. coli extract again. And the imaging was done on a Typhoon multifluorescence scanner. Yeah, CleanGels. Why would you need CleanGels? I just list up for you what is special, why CleanGels might be needed. So in some cases, you need acidic buffer systems. For instance, for membrane proteins, they are very often very basic and hydrophobic, and they have to be positively charged for a proper appropriate separation performance. But the casting and polymerization of acidic buffer containing gels is very, very problematic and difficult. And here you can use a ready polymerized gel, which has been dried down, and just soak it in the buffer that you need for the separation. I will show you an example soon. Then, if it is necessary to have gel buffers with a high pH value above pH 7, these gels have a relatively short shelf life because of alkaline hydrolysis. But here you can soak the gel with this buffer and get a better separation. In some cases, as already discussed, there is you need the presence of urea, and then the buffer, the urea has very short shelf life when it is in solution. It degrading to isocyanate. So also here you can make gels available very simply, ready-made gels soaked in urea. And this is the case for some native gel applications. You want to perform a cyanogram detection where you use biological activity of special enzymes for the imaging or for the detection of the proteins. And we have observed that the remaining monomers of acrylamide in the polyacrylamide gel in the ammonium persulfate can disturb some of the reactions. So we made some experiments after washing the gels and rehydrating the gels. The cyanogram detection works as well as in agarose gels. So I think this can be an important aspect. So how would you work with CleanGels? You place the gel onto the rehydration buffer with the dry gel side down. If there are any air bubbles, you just lift it up 1 or 2 times, so you can move the air bubbles out very easily. About 1 hour later, the gel is ready for separation. You just dry the surface with a sheet of filter paper, and then you start the run. You can get different buffer kits from Serva. There is a native buffer system for native cathode electrophoresis. So then you have Sortex kit. This is for particularly serial cultivar differentiation, where you are using the basic membrane proteins for differentiating the cultivars of barley, for instance, or wheat. And you do this in an acidic buffer system. And then there's the SDS buffer neutral or SDS electrophoresis with a high resolution. Here is an example for the cultivar differentiation, for quality control, for control of the suppliers for malteries and breweries if they have a mixture of summer or winter barley. And this special variants, these cultivars, can be that easily identified and detected by their specific pattern. For these, usually gels with 52 sample wells are used, that you have good statistical overview. And here you need fast chromacy staining. This is a staining which works without any alcohol, because these are alcohol soluble proteins. They are extracted with alcohol. So if you try to stain them with an alcohol containing solution, you would just dilute them from the gel. It's of course not very good to do that. And then we also have, for DNA fragment polyacrylamide gel electrophoresis, we have 2 choices: the Dilect buffer, which is usually directly used in the gel, gives very, very high resolution, very sharp bands, also for small fragments, and then a DNA disc electrophoresis buffer, which can be used as native or denaturing DNA electrophoresis. And here you see the gel that we can supply with the most sample wells, namely 103. This has here is being used for DNA fragments, quality control of DNA synthesis, and but this can of course also be used for proteins. Here detection was performed with silver staining of the DNA fragments. Now to isoelectric focusing, which is, as you know, of course, a separation according to the isoelectric points, and there are some specialties, important parameters in isoelectric focusing. First of all, you want the gel with large pore sizes, because sieving is not wanted. In isoelectric focusing, you only want to separate according to the isoelectric point, so you don't want the influence of the size of the molecule. And these 5% gels have large pore size. For them is of course very useful when they are polymerized on the film support, because they are very brittle, usually not easy to work with. You want to have high voltage, because close to the isoelectric point the net charge is very low. That means you need high electric field strength, up to 300 volts per centimeter, to get sharp focused bands. And that means for, like, say 10 centimeter separation distance, 3000 watts. You can't do that on a horizontal gel. You never can do that on a vertical system. The temperature has to be very accurate because the pK values of the samples and of the carrier-ampholites, the pH gradient, making reagents are highly dependent on the temperature. So a few degrees up or down can cause quite different pattern and isoelectric bonds. Sample application is also important because particularly under native conditions you need to find the optimal pH position, and this is inside of the gradient. So in a vertical system you can only apply at the basic or at the acidic end, but in a horizontal, no problem, can choose the optimum sample application point. And as I said already earlier, you might need urea in the matrix, 7 or 8 molar urea, and then you can easily achieve denaturing conditions in these gels. Here you see one example of the pre-coat, these very, very thin gels I told you. Here, of course, you want to have highest resolution, and you can use them for quantification of pork meat mixed in beef meat. So this is a thing which is, for instance, applied in food analysis. This is a very accurate method. You need a densitometer for this. The focus gels have also some specialties. There is, for instance, a focus gel 24S available, 24 sample wells, where you can apply the sample into the slots. That is making life much easier here. These slots take up to 24 microliter sample, and this is making life much easier than using sample applicator pieces or these sample applicator bands with the holes in it, the strips, the silicone rubber strips, because they are often leaking. Here we apply the sample very, very simply into the samplers. And as I said already, no need glycerol or something to make the sample more heavy than the buffers. And here you see here in the moment the electrodes are applied on the edges of the focusing gels, and here no IPG strips or no wicks and nothing is used, just apply them directly on the surface. Also makes it easier. Here is a result, an example for isoelectric focusing of oligoclonal IgGs for this in a clinical application where you need very high resolution and very high staining sensitivity. Here the serum of a patient is run, and this is the solution, the brain solution. Here you see some special additional bands, also recognized by these very basic bands, which can indicate a disease like multiple sclerosis. The sensitivity of this ammoniac-calic silver staining is very sensitive. Also for this there is a ready kit available which can be used for this. It's the best you can do for the most sensitive staining for isoelectric focusing gels. I forgot the name. It is cerebrospinal fluid. Cerebrospinal fluid is separated next to serum. Here are the Serva blue strips for 2D electrophoresis. They are freeze-dried in under vacuum, and you see we have these are 3 millimeter wide, and after rehydration 0.5 millimeter thin gels, and they are available with sizes from 7 to 24 centimeters, and in different gradients, very wide gradients, very narrow gradients, and also in nonlinear wide gradients. And now the final applications are the 2D electrophoresis, where you can achieve the highest resolution for protein analysis. And I show you here an picture from a very old paper, 20 years old, from Angelika Gert's group in electrophoresis. And here vertical, the result in a vertical gel is compared to a result in a 0.5 millimeter thin gel on a multi-fold cooling plate, and this sample was the same, the first dimension was the same, but the staining was the same, was silver staining. But you see immediately that with the thinner gel on the horizontal plane, you get much sharper spots and you find more proteins. And not only for detection, but also in mass spectrometry, you have a benefit there because the protein spots are much higher concentrated, so the enzymatic digestion is beneficially influenced by this. You get a better, more protein, more peptides out of it. For multiple runs, we have introduced the HPE Tower quite a time ago. There you can run up to 4 gels at the same time. Here you see the electrode lids. They're still here in the parking position, and these cooling plates are aluminum oxide ceramics plates, and they are connected for the electric supply and also for the cooling in one box. And if you wish, you can also choose to have one chamber just for one gel together with the power supply, as I explained already before. But you can also stack these ones because they are all in this drawer system. So you can, for instance, if you don't want to have 4 gels run at the same time, you can run only 3 gels, for instance. So you can start with 1 and then put more and more and more on top of each other with this multi-deck concept. So why is it called HPE system, high performance electrophoresis? First of all, because of the relatively thin gels and the film support on the cooling plate, you get more and higher concentrated spots. The gels are produced with a for long shelf life, so they don't change their separation behavior, and you can achieve excellent reproducibility because they are made also with quality control and industrial way, so that is different from lab cast gels. I can tell you there are papers already around which have proved that. You don't need any glass plates and buffer tanks anymore, and no agar-rows to seal the strips in, so that makes the handling much, much easier than in a vertical boxes. And this is a non-fluorescent film backing. The gels cannot break, and you can scan the gels with fluorescence, different fluorescence wavelengths. Here you see electrode wicks. Here an IPG strip is 24 centimeters, and here the marker proteins are pipetted into a small slot, which is always in these gels, that you can always have a standard run with it. Here is an example stained with MS-compatible silver staining. That works also better in a thinner gel on a film support than in a thicker gel. We have discussed that already in the detection webinar, which you'll find also in the YouTube. There was comparison of a vertical and these HPE systems done in a lab in Hanover, at the lab of Professor Hans-Peter Braun. So the sample was the same. It was extracted from cyclamen tissue, and also the staining was the same. Even the buffer system was the same. But you see, the vertical gels don't by far not show the resolution and the sharpness of the spots like the flat top tower, the HPE Tower. That was a technical paper in the year 2013. Martin Moche and colleagues, and there you can find also statistics and all these comparison between the traditional, conventional vertical thicker gels and the horizontal gels, and these were the same areas of one gel shown from the vertical and from this thinner gels, and you see immediately that the resolution and the sharpness of the spots is so good that you detect many more proteins than in a conventional system. Yeah, that was what I wanted to demonstrate you. That I hope you believe me that the horizontal approach can have a lot of benefits, maybe also in your lab. And I thank you for your attention. And if you should have any questions, please ask them now, and I will answer them right away.